Hi, this is Eli Ochiltree of the SnoKing Beekeepers Association. It's March 19th. It's our monthly meeting, and we are blessed to have two, not one, but two speakers tonight. And the first one will be Jennah Kemp. Jennah Kemp and Alex Cummings are the people you talked to on the phone or maybe met in person at the WASBA conference last October that help you with the Healing Through Hives program. And Jennah has to explain the rest of it. It's how they help veterans network and receive support from each other and various programs through beekeeping, which is, as we all know, a very bonding experience. So, and then after she speaks, Rex Roberton will talk about his work with Optera bees with the UBeeO, the unhealthy brood assay. So Jennah's going to take about 20 minutes, and we can take longer if people have a lot of questions. I don't object. And this is a really nice program. You've been on it, I know, for 3 years already, been working on this program.
Yep.
Okay. So Jennah, you go ahead and explain.
Okay. Thank you, Eli. Thank you all for having me. As Eli said, my name is Jennah Kemp, and I am the manager of our veterans program at GRuB. And GRuB is a small nonprofit in West Olympia. We have a small urban farm. We do a bunch of different programming around growing food and growing community and growing people. So our mission is to grow healthy food, people, and community with roots in the land. We create opportunities for people to learn, lead, and thrive. And so one of our programs is the veteran program. And we have two main projects that we are currently doing all around supporting veteran farmers and beekeepers. But veterans have been a part of GRuB's work since the very beginning, and we've been around for over 20 years. We started building backyard gardens using veteran volunteers, building backyard gardens for veterans. That was our GRuB Garden project, which is still going. Um, and we also had a victory farm. So it was a farm led and run by veterans as just a way for veterans to grow food together and build that community aspect. Um, and now we kind of like formalized into what we are now with these two big projects. So we have our Shepherd project and our Healing Through Hives project, which I'll talk more about. But our main mission in the veterans program is just to support veterans who are reintegrating, so recently discharged or who have been out for decades. It doesn't matter. That transition out of the military can be really difficult. It can be very isolating. And so we just want to help bridge some of those barriers that are there for veterans, support them if they want to get into farming or beekeeping, help them get started with education, mentorship, resources, whatever it is they need to get to the goal that they have. And we work really closely with them one-on-one to get there. Why veterans? Like I said, veterans face a ton of barriers. Obviously, we know a lot about these senses of isolation, higher rates of homelessness, of mental health challenges, and suicide, and high unemployment. And so our goal is really to help just navigate some of these barriers with veterans so that they can overcome these really, really difficult challenges and their families as well. And in Washington State, we have so many veterans here. We have 8 military bases. There's a huge veteran community here. And so it's the perfect place for this kind of project. And why agriculture? Veterans are really interested in agriculture. There just seems to be an innate like connection between folks that go into the military. There's that sense of supporting your community, that sense of purpose that they get from serving their community, and agriculture can often serve as like another avenue to express how they want to be of purpose and serve their communities. There's lots of health benefits that come with it. It can be very healing, very grounding. I'm sure you all have experienced those aspects of beekeeping, the emotional and mental health connections that you get from keeping bees. It can be really healing and really grounding and just really reintegrate you with the land, with the community, with your family. And it is a financially viable career path. It can be. It's a little challenging, of course, as we know, and that's why we need as much support as we can to make it, a viable career path, but it can be if it's done right. So our Healing Through Hives project, it's funded through we have a federal grant through the USDA. The grant is through the Beginning Farmer Rancher Development Program, BFRDP grant. And it's a 3-year grant where we can support veterans that want to get into beekeeping or just add to their already beekeeping enterprises or add beekeeping to their existing farms. And so our goal is to increase veteran-owned and operated apiaries in Washington State. And this year is the last year, but we are applying for an extension for another year. So we're hoping, but of course, lots of things are up in the air right now and this year, but we'll see. And our main partners for this project are WASBA, the Washington State Beekeepers Association, and the Washington State Farmer Veteran Coalition, which is another nonprofit. They're nationwide, but our Washington State chapter helps support all veteran farmers in Washington State. And if you haven't heard of them, I recommend looking them up. They do a lot of awesome work. So this is just kind of a brief overview of the Healing Through Hives project. So our 3 main goals with this project are to create beekeeping pathways and training for veterans. So by the end of this year, we just want to support more military veterans to get certified as beekeepers, to get the training and support they need to start beekeeping and to start a beekeeping business. And to do that, our second goal is to build a network of community apiaries for veterans. We already have 3 apiaries right now. We're about to establish another one, and we're still looking for a 5th location to start up this year, but I'll talk more about that. And our third goal is to support veterans through mentorship. So be able to connect beginning veteran beekeepers with mentors that are in their area or who specialize in certain aspects that that veteran might want to learn more about, and just be able to make those connections so that a veteran can learn from somebody with tons of experience and not have to make all the same mistakes along the way that a lot of beginning beekeepers have to make. Because as we know, it's quite a journey to get to that point where you really feel like you know what you're doing and you're comfortable, and just having some guidance along the way can make a huge, huge difference. So more specifically, what we offer for veteran beekeepers, we have funding through our grant to reimburse veterans for the certification courses through WASBA from beginning to journeyman, workshop fees, any other training or networking opportunities. We can provide like up to $200 of reimbursements for a lot of those opportunities that come up, including if you want to go into a master beekeeper course. And then we can also support you with like mileage and travel costs. So if you have to travel to get to these workshops or for these training opportunities, we can help cover some of those costs. The conference fees. So for veterans that want to attend the WASBA conference, we can cover those fees. And then we also have some funding to help provide like adaptive gear and supplies for beekeepers. So especially beekeepers that might have a disability or just need some extra supplies to kind of help them get started that fit their needs. We have some funding for that. And then we also try and provide hands-on experience through our apiaries that I mentioned. And at all of our apiary sites, we provide workshops throughout the year that are free for veterans to come and attend. We try to focus on, seasonal subjects that make sense, and also things that can specifically help them build their businesses if they're interested in starting businesses. So kind of the business aspect of beekeeping and like value-added products, things like that. And then mentorship and community building. So a lot of our mentors work one-on-one with the veteran beekeepers, like at our apiary sites, doing workshops. But then we also try and have events throughout the year just to build like that sense of community and networking. So veteran beekeepers can meet each other, and non-veteran beekeepers. We want to kind of bridge that gap so that veterans and non-veteran beekeepers can get to know each other, build those relationships. And so we have some networking events throughout the year at our different apiary sites. And then we also have some funding to support mentors. So if you are a mentor or somebody who might be interested in mentoring a veteran beekeeper, we have like small stipends to support you. We can also pay or reimburse for like some of the mileage or travel costs if you have to travel to mentor somebody. And then every year we host a peer support training. It's through a wonderful course given through one of our partners is an organization called Growing Veterans. They're up in Linden, and they offer this peer support training, and it's really geared towards folks that want to work with veterans, just to learn like the specific challenges of working with veterans, just to help like prepare you if that's something that you're interested in. I've taken the course. It's actually I recommend it to anybody. It's really awesome. And then what Alex and I provide specifically is one-on-one support with these veterans. So when a veteran reaches out to us saying, hey, I have no experience, but I'm really interested in beekeeping, we will sit down with them, we'll learn what they're looking for, what their goals are, and try and just connect them with the specific resources they are needing so that they can reach their goals. And we work with them throughout their journey. Who's eligible? So military status, we will help support military veterans, of course, active duty as well, and we do support spouses and dependents as well. And this is a Western Washington only project. So we are restricted to Western Washington, unfortunately. But through our other project, our Shepherds Project, which is funded through a different grant, and that is a statewide program, we are able to also support beekeepers in Eastern Washington, but just with a different funding source and with some different restrictions around it, but pretty similar. And we've already been working with an apiary up in Spokane through that project as well, working with Ellen, the vice president of WASBA up there. So we really can reach all throughout the state. And we try and keep it really low barrier. So we We don't ask about your discharge status. We don't even ask for proof of veteran status. Yeah, we just want to support as many veterans as we can. And this is some of our other partners. So in addition to WASBA and the Farmer Veteran Coalition, we have some other partners who can also support our veterans. So I mentioned Growing Veterans. They're the folks that do the peer support training, but they're also an awesome farm up in Linden that veterans can come and just get their hands in the dirt and grow food together. So they're great. And we also do partner with the WDVA, the Washington Department of Veteran Affairs. So if there's a veteran that might be interested in getting like a claim going to get into their VA health services, we can help connect them to like a veteran support officer to help them get their claim done. And kind of just help them through that process because it can be really intimidating and drawn out and difficult. So it helps to have somebody who knows that process really well. And then through WSU Extension, they have a great farm planning course called Cultivating Success. And we've worked with them. A lot of our veterans have used that to build farm plans. And then we've also partnered with the Thurston Conservation District because we're in Thurston County, and they can do like provide technical assistance for any of our veterans. So they'll actually come out to their land and help them do like a planting plan for their farm, or just help them kind of plan out how to plant specifically for pollinators. And what kinds of plants and where, and kind of work with their land. And then they also provide workshops that are free for veterans through us as well. So these are some of our apiary sites. As I mentioned, a big part of our project is having these community veteran-focused apiary sites where veterans can come and get hands-on experience, not have to invest in getting their own bees right away if they're not ready. These are spots where they can come and still work with bees if they're not ready to make those huge investments right away. So some of our sites, we have one at the GRuB farm in Olympia that we just established last year. And we also have one at the Ording Veteran Farms in Ording. It's a really cool farm. It's an incubator farm for veterans. It's supported through the Washington Department of Veteran Affairs. And they have an incubator farm where veterans can lease like quarter-acre plots for very little. It's like $50 a year, and use those spots to farm and do any kind of farming out there. And so we have an apiary site there, so those farmers can also learn about beekeeping. And the farm manager is a very, very experienced beekeeper. She's been beekeeping for 30 years, so she's really awesome. There's also some hives out at the Veterans Ecological Trades Collective, which is another nonprofit in Rochester. And they have just a huge 120-acre plot that's just wide open, lots of wildlife. It's a great spot for kind of our example of like commercial beekeeping. Alan's been keeping, he takes out a bunch of his bees out there every year. And yeah, that's been really great. And then this year we're going to be adding an apiary at the Roots Farm, which is in Paulsbo. They work with a lot of veterans there. They have interns that are veterans who are really interested in learning about beekeeping. And so we're going to be setting that up in the next few weeks. And then we should be also putting some hives out at the Growing Veterans Farm in Linden. And we do have a very small apiary at the governor's mansion as well, just 2 hives. But that's been a neat partnership working with them just because they're so close to us. GRuB has also built a little community garden space there at the governor's mansion, so we can do events and workshops and stuff out there. And in addition to everything else, we really try and focus on a holistic approach when we're working with veterans, just because of their specific challenges and barriers. And so we try and also really focus on like the mental health aspects, PTSD support, suicide prevention, the peer support that I mentioned, and that aspect of just community support. So building those connections and relationships so veterans don't feel like they're going at this so alone. Really makes a huge difference. And how can you help? If you're not a veteran, obviously if you are and you want to take advantage of some of these, please reach out to me. We would love to work with you. But if you're not and you're still interested in supporting veteran beekeepers, firstly, spread the word, which Eli is helping us do right now. But yeah, through WASBA, we can reimburse folks for all of the certification courses that are done at any of the clubs. But a lot of clubs just don't know about it. And so we're trying to spread the word that way. So if you know beekeepers at other clubs or in your own club, like let them know that these resources exist. Let them know about us. If you know veteran beekeepers, please send them our way. Another great thing you could do is be a mentor. Like I said, we have ways to support you with that. So we have stipends we can pay. We can reimburse you for some of the costs if you have to travel. We'll support you in getting started as a mentor, help connect you to a beginning beekeeper. So that's something we're always looking for. We really want to try and find mentors that are throughout the state so that when we find a beginning beekeeper that's looking for a mentor, we know somebody that's close to them. And if you're interested in or close to some of our apiary sites and you just want to get involved there, we would love that as well. We hold workshops at all our apiary sites. So if you would just want to like teach a class or come out and help do anything really, I mean, all of our apiary managers are super busy, so I'm sure they would love any support. And just come out to our events and connect with our veteran beekeepers. And that goes along with training education opportunities. So if you have something you really want to share with our community, reach out to me and we can work on how we could have you come out for a workshop or even do like a recorded thing. Yeah, just let us know. Partnerships, we're always open to anything. Like I said, this is our last year for this particular project, and we're starting to explore how we can continue to support veteran beekeepers if we aren't granted an extension or if this grant ends for whatever reason. So if you have some ideas, you can always reach out to us. And donations are always welcome. GRuB is a small nonprofit, so we always accept donations and appreciate that. And that's all I have. Are there any questions?
Thank you so much, Jennah, because it's overwhelming for us. I'm sorry. I tell you that every time and you explain it very patiently. But okay, I wrote down where the apiaries are. Did people hear apiaries that they might be interested in calling Alex or Jennah and saying, I'd kind of like to meet veterans out there, or I am a veteran, or I'd be willing to come out and talk about a class? Because that's a pretty big spread. Okay, Paulsville, Olympia, Ording, Rochester, Linden's just a few hives, right? And then the governor's mansion is a few hives, but that's a pretty big spread of
Yeah.
area. And if you don't know where those places are, like Rochester, Rochester? But yeah, I know. It's like I grew up in Tenino. People have to look it up. Where's that? So, but yeah. So yeah,
And
I
I'm
should
adding
look
my
in the chat.
my email to the chat, so
Yes. And
feel
it's
free.
easy because it's Jennah@goodgrub.org, right?
Yeah, or
Oh,
we have
veterans. Okay.
a a generic one. Yeah, I'm sorry, I just shared that with you. Let me do
And
that.
and and the people will probably be talking to you or Alex.
Yes,
And if
we're a
you,
small team, just the two of us.
yeah, and if you need receipts for past years, I do my best to keep track of everything. And if I dig hard enough, I can find proof that you took a class, 2 years ago or so, and this is a 3-year grant. So can you go back kind of
We can't go
year
back within our
two?
fiscal year, so we go back to June. That's
Okay.
as far
Okay.
as
But
we can go,
quick before
unfortunately.
you may finish the certificate in June. And the conference would be great too. That's going to $200 this year. And
Yeah.
I really think I saw you and Alex both there at the table, the grub table. I don't know how many people realized you were kind of around the corner, but are you going to be there again this
Yeah.
October?
Yeah.
Okay. I really think it'd be neat if all the veterans kind of got together at some time, either Saturday night dinner or for breakfast Saturday morning, or,
Hmm.
we put up a little
Yeah.
sign, anybody who'd like to sit over here with the vets. I mean, wouldn't that be just neat? I think that would be so neat. I was going, hey, we got all these people here, and I know some of them are vets. That'd be kind of neat to say hi.
That's a great idea. I love
I mean,
that.
we say thank you for your service, but like you said, we need a little more. I mean, it's important. So all you vets, thank you for your service. I appreciate it. My dad was in the Navy and I understand. And we moved around as a military family and settled in Tenino when he retired. So,
Hmm.
so I grew up in Tenino. Where's Tenino? But and a lot of vets come back to this area, an incredible number. That's why it's 10% of the population.
Yeah.
People
Ton
come
of them
through those
end up
8
staying.
military bases and they like this place. And then when they get settled, yeah, it's time to get bees, right? As soon as you buy land,
Yeah.
everybody needs bees, right?
So
No, actually, one of the neat things about the apiaries, anybody who's hesitating to go out to one of those, if you know any vets who don't know if they want to keep bees, the real test is you dress them up in a bee suit and the gloves and everything, and they stand at the hive. And if they hold that frame of bees and go, wow, that's when you got them hooked. That's the people who should keep bees. If they want to turn around and run, that's okay. But they're
and
probably
that
not a
happens at
beekeeper.
almost every event we do, our community building events. There's folks that have never been
Hmm.
around a hive before. It's their very first experience, and you can just tell as soon as they open up that hive or pull out a frame of bees, and they're just hooked right away. And we've actually had folks from those events go directly and like join their closest beekeeping club and start beekeeping.
Hmm. And if you don't get renewed, it's kind of important that everybody know where these veterans groups have been meeting. And then because we'd like to stay involved, all of the member clubs really would like to stay involved. Do you have a
Yeah.
newsletter or people still have to call you to kind of find out what's going on at each apiary?
GRuB has a newsletter. You can sign
But
up through our website. It just covers all of our activities, and anything that's going on with the veteran programs will be on there as well. And on our website, we have like upcoming events, um, things like that. So I can share that as well on
yeah,
here.
who
It's
to
good.
contact? Because
Good
sometimes
for
people
us.
are hesitating and, you know,
Yeah.
I just feel like sometimes we need a little nudge,
Mhm.
we care about you.
Yeah, if you come out to an event or you sign up or show that you're interested, we always send out emails,
Mhm.
invite stuff to all of our workshops and events. Well, I guess there's no other questions.
Yeah, I don't see in the chat. Let's see. And we know how to find you, and you've been here before, and this is recorded. So
Mhm.
thank you so much for coming. We really appreciate you so
Oh,
much
my
and what
pleasure.
you do. And I hope you do get a renewal year, but if you don't, want to make sure we see you at the WASBA and sort of make plans for keeping everybody in the loop.
Yeah, I
Yeah.
won't
And
unfortunately
you
be
will be
there,
too.
but Alex will be there at the
Okay.
conference. I'll be on maternity leave, but
Oh, congratulations. Wow. Okay. Yeah. And GRuB is not going away. You're going to
no,
be around.
we've
Yeah.
been around a long time.
It's
Yeah,
just that the 3-year grant might start running out.
yeah.
So,
But we'll pivot. We'll figure out a way to keep supporting veteran beekeepers.
and let us know what we can do to help. Okay.
Yeah, absolutely. And please, anybody reach out to me if you have any
Mhm.
questions or just ideas. We're all
Okay.
open. Lovely to meet you all. Thank you.
Thank you.
Bye.
Okay, Rex, you're sharing
Yes.
screen next. And Rex, I sent out your massive bio, and now my mind is blank, except that you did this really neat thing that you're going to explain what you did working with Optera bees on this unhealthy brood odor assay, right?
That's it.
So I hope people read it. If they didn't read, it was incredibly long, all the stuff you've been doing. But we sort of knew that because you already did a presentation for us about that idea of queen isolation for Varroa control. And it's back like a 2021 monthly meeting recording. It's still there on YouTube if people look for it. Look for Rex Roberton.
Okay, Eli, are you seeing
Yep,
my
seeing it. Looks good.
presentation full
Looks
screen?
really good. So,
Also, can you see my cursor moving
yes,
around?
and your cursor is visible. Very good.
Can. Good,
Okay,
because I like to
ready?
use it as a pointer.
And I'm kind of excited about this.
So,
I have to say that this and going to the SBGMI's meeting, oh, Colleen was there too. I think David made it too. They were talking about the Harbo assay and their queen programs, which are different. I know they're different, but they said they had a video on how to do the Harbo assay and that it really wasn't that hard, which is what David, who's on here, David H, keeps telling me. It's not
yeah,
that hard. Okay,
David
this
does it.
sounds even
Yeah.
easier. And like a
Mhm.
beekeeper could really use this. So I better stop talking and let you get going. Okay, thank you.
Yeah. Okay. And are you still recording?
Yes.
Okay. All right. So yeah, let's get started. So I do have to do a little disclaimer. I don't have ownership in Optera. I'm not an employee, but I guess you could call it a consulting product designer, and I supply the precision guide and the test ring. So that's the conflict of interest that we have to get out there. So a quick overview. I'm going to do a real short introduction. Then we're going to talk about kind of comparing the different hygiene assays. And then we're going to talk about the UBeeO pheromone and then the UBeeO assay, and then mechanisms of resistance, and then how fitting it into a management program, and mostly how I fit it into my program. And then I have a bunch of resources at the very end that for anyone that wants to do some more reading and kind of expand on what I'm showing, because there's a lot to it. So like just real briefly, the UBeeO assay is a pheromone. It's a product that you buy and you spray it on a section of the brood. And so you can see this group of bees on this comb here, and those are nurse bees, probably like they say age 15 to 20 days old. And 2 hours prior to taking this video, I sprayed the pheromone on a little section of the comb, and the bees think that there's something wrong because the this UBeeO pheromone is a general stress signal. So they're actually going in there and opening up those cells. And if the bees were not hygienic, they would totally ignore it. There would be no cells open. They wouldn't even know it's there. And uh, so it's a spray. You put it on the brood comb. It's 2 hours, and then there's a way of scoring, and we're going to go into that. And then a little bit about how I keep bees. I usually have 8 to 12 colonies. And I raise my own queens every summer during the honey flow, so June and July. And so every year I have to decide which queen or queens I'm going to raise daughters off of. And for me, this UBeeO is a game changer because up to last year, I've just had to guess about the hygienic level of the colonies. And I knew that I probably had hygienic traits because the original bees 7 years ago were from Dan Harvey, and he started with feral colonies in catching colonies out on the coast, and then bringing in SMR stock and Russian bees and some different stock. And then he's never really used chemicals, so it's like, you survive or else you die. So he did the work. I knew I had the genetics there. A lot of times when people go in with the UBeeO in an apiary, if there hasn't been any selection, then they don't have any response. It's basically 0% of hygiene. So now with the UBeeO, I have a way of scoring these colonies. And since I only do like 8 to 12 colonies, I can score every queen. And I'll kind of get into that, how I do it. I do it twice a year, like new overwintered queens and then new queens in the fall. And that's kind of the gist of it. So let's talk about the hygiene assays and different ways of measuring. So Marla Civic back in the 90s started perfecting this freeze-killed brood. And she did a lot of really important work with this. And the important thing to note or remember about freeze-killed is that it's a selection for general hygiene. It's not specific to Varroa, and you don't end up with like high Varroa resistance by using this selection process. Now, if you use it to select for a few years, you'll be more likely to have some Varroa resistance in there. But it's different because the bees are picking up on this oleic acid, which is a general insect necromone from the dead pupa. And it's a 24-hour score, and then 95% removal from the test area is considered a high score. Then Harbo, also back in the 90s and early 2000s, developed the SMR, what he originally called the SMR, which stands for suppressed mite reproduction. And it's a way of quantifying the non-reproducing mites. And you get a score, and the score is like 1 to 4, and ideally you want, the high number 4, 3 or 4. And this is a very good way of scoring for hygienic behavior, but it's a little different than the UBeeO. So about 12 years ago, Dr. Kara Wagner started working on these, what she calls the unhealthy brood odor. So by extracting pheromones out of the head space in the cell, in that area where the pupa is, they were able to identify these pheromones. And it's not specific to Varroa mites. It's can also be stimulated by viruses and other infections. But you get a score from a 0 to a 100, and I'll kind of get into this. And the good thing is that it's more of like disease and Varroa detection. And this UBeeO odor or UBeeO is like naturally in colonies, and then as I was saying earlier, the test kit has a pheromone, synthetic pheromone that's in a hexane solution. So now in research, there's other ways to test for hygienic behavior. One of them is like measuring the infestation frequency in a highly infested comb. Then you insert it into a test colony. Then you re-examine a week later, and then you determine the infestation frequency again. And then there's a way of scoring that. Reduction in frequency is a measure of varroa-sensitive hygiene. Then there's different ways of measuring the percentage of cell cap removal. And so that's been around. Some research they'll actually insert mites into cells, cut a little slit, throw a mite in there, and then measure the rate of removal. And then there's a pin-killed assay. The thing to remember is when someone says these bees are VSH or they're hygienic, or if you're reading research and they're talking about VSH or hygiene, you want to, hopefully they're telling you how they measured that, because these measurements are different, and it might mean something that's you might not be expecting it to be meaning. So if I say UBeO, I'm talking about the pheromone. It's an unhealthy brood odor. And if I say UBeO assay, then I'm talking about the product that you use to score this hygienic behavior. So a pheromone is usually a group of different molecules. Sometimes they'll say it's a bouquet of different molecules. It's very rare to have a pheromone that's just one single molecule. And the UBeO is a group of different pheromones. And we probably all know that the definition of a pheromone is a chemical substance that is produced by an animal to communicate with another individual of the same species. And in the case of UBeO, the pupae are signaling their compromised state to the nurse bees. So we have a signal transmitter and then a signal receiver. And so what is the message that the pupa is sending? She's saying, something is wrong, come and check it out. You may need to sacrifice me for the well-being of the colony. So it's a stress pheromone, and it attracts the nurse bees. And the message actually is, you may need to come and kill me and haul me out of the colony. So there's specialized cells in the fat body tissue that produce the UBeO. So these enocyte cells that are in the fat body tissue produce the UBeO pheromones. They circulate into the hemolymph, then they accumulate on the cuticle or the epicuticle, which is the outermost layer of the exoskeleton. So because they accumulate on the cuticle, they call them cuticular hydrocarbons. You'll see that in some of the research papers. And they're volatile, and they can pass through the wax capping. And UBeO is not specific to the varroa mites. I already said this. It's a general stress pheromone, and a high virus load can also stimulate the hygienic behavior. There's a really interesting study that was done actually back in 2018, and Dr. Wagner and Marla Spivak and Olav Ruppel did this, and they found that the brood actually affects the behavior of the honeybee. Previously, everybody thought like the nurse bees that are hygienic have this hypersensitivity to these pheromones, and non-hygienic bees don't have this ability. This study, they took what they call cross-fostering, where they take brood from different colonies, different types of colonies, and put it in other colonies and see what happens. And they found that the brood from the hygienic colonies was more likely to be removed than brood from non-hygienic colonies, regardless of where the brood was fostered. So that means these hygienic brood out of a hygienic colony, put in a non-hygienic colony, the quality of that signal that that brood is sending with the pheromones was enough to actually stimulate non-hygienic colonies to inspect and open up these brood cells. So hygienic related brood signals complement previously identified characteristics of hygienic adults. So that kind of goes into, the UBeO and the signal sending. Now we'll talk a little bit about the signal receiver. And of course, that's the antenna on the nurse bee. And the antenna has a bunch of different types of what we call sensilla. And this illustration here is what they call a sense plate. And this is where the pheromone is picked up and the signal is received. So electron microscope images of the antenna, so these oval circles here, these are the sense plates, but there's also other types of sensilla, these hair type shapes. In the middle right here, there's these like volcano type things with an opening. Over here, there's like a couple of, there's a depression, then a hole, and then kind of a knob down in there. And these all have different purposes, sensing humidity and different things that the bee needs to know about. So in this image in the foreground, you can see a couple of these sense plates. So you can see kind of a raised ridge, and then there's a channel all the way around, then a rim inside, and then it's dished out into this dish shape. And there's let's go back here so you got these openings here, these pores where the UBeO can come in. This is that little ridge and the channel, and then the ridge around the inside. And then they call this a cap cell, and then inside is a nerve cell or a sense cell. So there's a nerve body, and the dendrites extend into the cap cell. There's a fluid in here, and then the axon goes out and connects with, actually there's thousands of these that connect, and they go back to the antennal lobes in the brain, and then they're processed, the signals processed there, and then it goes up into the what they call a higher part of the brain, the mushroom bodies, and then somehow it stimulates this hygienic behavior. So in this fluid that's in the cap cell, there's odorant binding proteins, and it seems like this is the thing that is specific in hygienic bees. They have the quality of these odorant binding proteins, their presence, and the way the specific ones that they have, they are able to they pick up the molecules of the UBeO pheromone, and they carry it over to the dendrite to a site, and that stimulates the nerve. So a chemical signal is converted to electrical, and it goes back to the brain. So so what does it mean for a colony to have a high UBeO score? So this graph is from one of the studies that Dr. Wagner did back in 2021. And along the vertical here, this is the mites per 100 bees. So 10, 20, 30, 40 mites. So this is your mite wash. And then as you progress along the horizontal, this is the UBeeO assay score in a percentage. So you got 0%, 20, 40, 60, 80, and 100%. And what they found when you get up to this 60% range, these colonies manage the mites on their own, and they don't need chemical treatments. So over here on the left, there's a list of some of these things that they're starting to discover with all this research that's being done. So the mite level is below an economic threshold. Winter survival in some of these studies was 41% greater compared to low UBeeO colonies. There's a study that she did on memory, 24-hour memory, and they did better. The UBeeO, high UBeeO, did better on memory. These colonies have a lower virus load. And then it's interesting, even the mites in the high UBeeO colonies have a lower mite load compared to a lower virus load compared to the virus load in mites that are in non-hygienic colonies. And there is a question mark here because this is, I think, so far just one study. So it needs probably, more work, but that's an interesting study. And I think it's same with this study on the gut microbiome. That's an early study too. They have found that there's less nosema in these colonies and less chalk brood. Now, I don't think anybody has done studies yet on American foul brood or European foul brood, but I think it's highly likely that they're going to find the same thing because if you look at the early research going back into the 1930s with Park and into like the 50s and 60s with Rothenbuehler, they actually did the earliest studies on hygienic behavior, and they developed stock that was had hygienic behavior against the American foul brood, and it was Rothenbuehler, I think, in the 60s that showed that there's these genetic traits that bring this hygienic behavior about. Okay, performing and scoring the UBeeO assay. So I'm just going to kind of touch on this because there's a lot to it. But if you go to the opterabees.com website and on the navigation drop down menu for testing resources, select UBeeO testing resources, and it'll take you to a page with a hyperlink, and you can download this manual, and it's done very well. It's like something like 26 pages, and everything you need to know is in there. So just touching on a few of the things, when you do the scoring, it's best to do it during a honey flow. And so a lot of people are doing it, in the spring, once the flow is going. There are environmental things that will affect. You can have a colony that is scored hygienic. They are hygienic, but there may be, if you test them at another time, there may be variation in that score, and these are things that they're still trying to figure out with research. But it's recommended there's the honey flow on. If there isn't, then start feeding syrup ahead of time to kind of stimulate the bees. You want the colony to be queen right for at least 7 weeks because you're testing that queen's nurse bees. So if you put a queen in a colony and then do a test the next week, you're not testing that queen, you're testing whatever nurse bees are in there. And if you put a colony together from several different colonies, you're not going to get a reading off of that queen. So 7 weeks of brooding for that queen. You want the test brood to be in the purple eye stage or younger. If they're older, the UBeeO spray with the hexane, it can stimulate bees that are ready to emerge that day. or within a day or two, they might start emerging out on their own, and not as so cells are being opened by the bees that are emerging rather than the nurse bees opening cells to inspect. So purple stage or younger. And you definitely want to use the spray guide and the test ring when you do this, because it controls the dose very precisely. And before we had the spray guide, you were kind of doing your best to hold it at the right height and keeping it vertical. And there was a lot of variation when you did it that way. So you're going to, at time 0, you take a picture of your test area, you do your dose, and then you, and it's 3 doses of this spray. So you spray, wait 30 seconds, spray again, wait 30 seconds, spray again. And then you do a photo, then you put it back in the colony, you wait 2 hours, and then you pull it back out and do another photo. And that's how you do your scoring off of those photos. So you can actually calculate your scoring later. You don't do it right then in the apiary. And then of course, there's a formula. So the device, the applicator looks like this. It's got a spring that you compress to get the mist to come out. And then as you release it, the handle pops back into the preload position. So I'm just going to say a couple pointers about this. Like I say, you really need to go into the manual, but it's important to make a quick compression so you get a fine mist. If you squeeze the handle slowly, you get a little squirt, and that's not going to cover the area, and then you're going to get a high dose in a little area. So you do a quick compression, and then you want to let that handle come back slowly, because if you let it slam back real quick from that spring, sometimes it pulls in too much. It pulls in air and you get a big bubble. And this picture here, this is the applicator barrel, and you can see this big bubble in here. And so I had some trouble with this at first, and I just trained myself to look at the barrel every time and see what the size of that bubble is. It should be more like a relative size like this. And this barrel, like there's a stainless steel shaft here, and this is an O-ring here, and your dose is in here, and it's pulled out of a vial that's up above here. And when you do that quick compression, this gives you an idea. It's only 0.167 mils, but it goes into a very fine mist. And so the test ring sets down, you set it down, there's a little ridge on the bottom of it, and you make a slight depression carefully. So when you come back in 2 hours, you know where to put the ring back and get your photo. And then the precision guide, it goes over the test ring, and then you'll want to support that precision guide with one hand and then do your compression with the other hand. When you compress that handle, it makes a kind of kicks the nozzle to the side. And so you want to kind of stabilize it and then do your quick compression. So your test area, that's the starting time, of course, has these cells that are undisturbed. And then 2 hours later, if you have hygienic bees, they'll have cells opened up. And then so you take a picture again. Now this colony was actually breaking up the larva and pulling it out to haul it out of the hive. But the bees don't always do that. This colony here, they just opened up the cells, and then they put their antennas in there to check the smell and detect the health of the bees. And they actually don't disturb the pupae. And other bees will come along and recap these. So the way I score it is I just quickly make a pencil drawing. So the cells are in a row. So I just, like the top row here, there was 4 cells in that one. There's 5 in this, 7 in this one, and so on. So I totaled up the number of cells that I had to start with. Then I go in with a marker and I mark the ones that were opened up. But I don't count those. I count the ones that were not disturbed. So I have 1 in this row, 3 in this row, 2 in this row, and so on. So 13 altogether, and then you punch that into your formula, and that's how you get your score. So this colony is highly hygienic, 71%. So that's kind of the basics on the testing. Let's talk a little bit about mechanisms of resistance. So we know that all around the world, there's these populations, feral colonies, that are hygienic. They don't need any treatment. They've adapted. It seems like it takes about 5 years or so for a population to recover. And, the process, of course, is that the mites come in and kill off most of the colonies, but there were some colonies that had the expression of these traits present at the time of this, assault, and they survived, and then they were able to progress, multiply each year. So Grindrod and Martin did a study. They titled it "Parallel Evolution of Varroa Resistance in Honeybees, a Common Mechanism Across Continents." So most of these colonies or these populations have been studied, and they collected all of these research papers on these feral populations that are resistant. And they found that there are 3 traits that are common in all of these. The first one, they call it recapping, which of course is the UBeeO sensitivity, where the bees detect something is wrong, and they open the cell up, and they either recap it or they remove the brood. And the third trait was that they found that there was the reduced mite reproduction. So these resistance traits suppress the reproduction of the mite. Now, I actually haven't quite finished that article, but it doesn't look that Martin talks all about grooming. I've heard some researchers say that grooming is always a part of Varroa resistance. Now, I'm not sure about that. I do know that Renderer, when he was doing the years of research that he did with the Russian bees, he found that whenever, let's put it this way, the higher the VSH score was on the Russian bees, then the higher their scoring was on grooming. So it seems like the grooming behavior travels with the what I'm calling the UBeeO hygiene, this uncapping, recapping response, those traits. But it's very possible that some populations don't have those together. Some might be stronger on grooming, and some hygienic colonies may not have grooming. I don't know. It's something that maybe I just haven't come across all the whatever information there is on that. But the result of this UBeeO hygiene, this uncapping, recapping, removal, is the Varroa mite ends up being in an extended dispersal stage, where they're not, their interrupted from their regular brooding cycle inside the brood. Other things that happen, like fewer multiple infested cells, lower proportions of infested brood, interrupted breeding cycles, reduced mite fertility. Whatever these things are, they add up, and the result is that the mites just can't reproduce enough to get above that economic threshold. But when you go into like looking at research papers on hygienic behavior and varroa resistance and feral colonies surviving, there's other stuff to it, and you'll come across things like resistance or tolerance to deformed wing virus, the timing of the brood production, reduced attractiveness of worker brood, something they call the brood effect, where the brood and possibly even something in the brood comb from hygienic colonies that can suppress mite reproduction. Renderer talks about that in his book, which I'll get to later. Van Alphen talks about this local population effect, where there is a high rate of resistance alleles in the population, and then something like absconding and frequent swarming, which we know, like the Africanized bees use that. Okay, so management strategy. So I'm just going to kind of do a little bit about how I'm going about this. My goal is no miticides, including the organic miticides, and to manage in the most natural way possible. So I've never used any of the synthetics. The only thing I've actually used is oxalic acid, but I don't want to be doing that either. So I'm working towards eliminating that. So really the fundamental of actually being, sustainable is knowing, doing your homework and knowing the bee biology and behavior. And, in the first year or two of beekeeping, you don't have that, but there's a lot of, reading and experience in the apiary that brings that. Trying to depend on one silver bullet is not going to be, effective for the long term. And, a lot of people, they've been trying to rely on amitraz, and now, the mites are becoming resistant. So that silver bullet is not working anymore. What I'm doing is combining these hygienic traits. So the bees have their own ability to deal with the varroa mite. And then I do a summer brood interruption in June. So the colonies all go broodless, and that knocks the mites out. It's very powerful for controlling the mites. And it's just copying what happens naturally when a swarm, a colony swarms and there's that broodless stage. So yeah, and June testing of all the colonies. So every queen that's overwintered tests those queens, and that helps me decide which ones I'm going to breed from. And ideally, I'd like to see them all hygienic so I can raise daughters off of each mother. And then in September, the end of September, the new queens can be tested again. And that's what I did last year. And then an important point is you have to do this every year if you're going to keep these hygienic traits, because it depends on the drone population. You could slowly lose these traits. So back in June 21st, my first experience with the UBeeO kit, I tested 5 queens, and I had 3 that scored low and 2 that were medium scored. Then I raised 5 queens off of these 2 higher scored colonies. And so over here in September 21st, it ended up being exactly 3 months later, I tested all the queens again. So the new ones, and then retesting 3 of these older queens. And so you can see they're scored here from the lowest to highest. And I had, so there's a question mark here because I kind of screwed up on the dosing in September on this queen. And so I'm just staying with the original score that I got, but that's what that question mark is. So I ended up with 2 colonies that were low, like 0 to 39% is low, 40% to 59% is medium, and I had 2 of those. And then I had 4 that scored high, 60 to 100% is high. And notice that 2 of these colonies that I tested in June actually scored higher in September. And I'm sure that that's because in September, I used the precision guide. So I had my distance on that nozzle tip exactly where it should be and exactly at the right angle. And when I used it in June, I was back a little bit too far and it kind of at an angle. So some of the spray, not all of the spray is going in, and some of the spray is hitting the side of the test ring. So my educated guess is that is why these scores came out higher on those 2 colonies. So on this queen, I ended up getting 4 daughters off of her. And note that 2 of them were low and 2 of them were high. And this is what you're going to see. You're not going to have a high scoring mother that always gives high scoring daughters. And this queen that scored 56% in June, I got a daughter that scored 71%. So these are my mite wash counts in September, well, end of August and 1st of September. So this kind of illustrates when I'm trying to decide what queen or which queens do I want to use to raise daughters off, using a mite wash is worthless for me. Because I'm doing these brood breaks that essentially wipe the mites out of the colony. And so I have to do, in order to identify hygienic behavior, I have to do this UBeeO scoring. Now, if I'd have been smart, like 7 years ago, I would have started doing the Harbo scoring. I wish I would have done that, but I just kind of, I didn't look into it. And I thought, wow, that looks complicated. But anyways, my point is, I need a way to score these queens, and because I want to keep this hygienic behavior up high, and the UBeeO assay is actually pretty easy to do. So so each colony last year had 1 dose of oxalic acid vaporization. That was their treatment for the year, as far as any type of miticides. So the queens that I've overwintered, I caged those in June when the honey flow starts. I cage them on 2 empty drawn combs on brood frames. So she has to stay on those 2 frames. She lays those up. The mites, and she has, there's no other brood for mites to go into. So any mites that are in the colony, they go into those 2 combs, and I pull them out and freeze them. So it's a 20-day caging, then trapping combs into the freezer, and then on day 25, I do a OAV. So day 25, all of the brood in the colony has emerged, even all of the drones. So every mite is, if it's not in those trapping combs, then it's in a dispersal stage. And that's how I get these mite wash counts in August and September that are 0 or 1 or 2. And the new queens, they don't need to be caged because they're going to be, that colony is going to be broodless because they have to raise a queen. Excuse me. They have to raise a queen. She has to mature. She has to fly, get mated, come back, mature a few more days, start laying. And then right before her brood, her larva is going to be capped, then hit them with 3 grams of OAV. And that's the treatment for the year. I'm not doing winter treatments or a spring treatment. So I want to make a point about, well, a couple of things, I guess, the importance of actually measuring your hygienic score and doing that every year and not guessing about, which colony is the best. If you have like, 5, 10, 8 to 12 colonies, maybe most of them look really good. And that doesn't mean that they're all hygienic. And I want to know which ones have these traits. And so this is just a kind of a hypothetical example of, if, say I took that, say I guessed and I accidentally picked the lowest scoring queen and then was unlucky next year and picked her lowest scoring daughter and did that a couple of years in a row, I could end up with no more hygienic traits or very low hygienic traits in the apiary or in the colonies. Of course, it depends on your drone population, but this is, just a kind of a visual to kind of illustrate that you need to be doing the selection and the scoring, and you need to do it, every year. And guessing is only guessing. Okay. So what about drones? Drones, question mark. So the bottom line is you have no idea what the drone population is. You're open mating, or I'm open mating. I do hope that there are feral colonies. This is Port Angeles here, and I am about 5 miles from the waterfront, up right next to the park, Olympic National Park. This is my place here. And back in 2018, I spent a year obsessed with beelining and bee hunting. And I found a feral colony about 1/3 of a mile away up in some managed forest land. And then right next to the park border, 1 mile away. So this is a radius of 1 mile on this inner ring here. And there was a feral colony there. And I also had a colony coming from 2 miles away. I didn't try to find those bees because, for one thing, it's 2 miles away, and then there's a lot of private property and a deep gorge and a creek that goes through here that you wouldn't want to try to get into. But yeah, so it would be nice if there's feral colonies that that my queens are mating with, but you really, you have no idea, and you just want to. It is what it is. This slide, my friend Greg Butler put this together a few years ago in the context of mite bombs, how easy it is for mites to spread from one colony to the next. And he put in the address of everybody in the bee club. This is Port Angeles over where the red circle is, and then this is Squim over here. So this is like 15-mile distance between these two. I think this is something like 86 apiaries. So you could literally have hundreds of thousands of drones in this area, and you don't know what it is or where the congregation areas are. So point being, test your colonies every year and see what you're ending up with. So let's jump to resources that are available. So I already mentioned this instruction manual, and that's definitely if you're interested in this and you're trying to figure it out, go and read the manual. And if you're going to be doing the UBeeO, definitely study this, because there's a little bit of a learning curve, and you want to get the details right, and do it as accurately as you can. You can go to Optera's website, and there's a drop down under testing resources, and you can find these abstracts to the research papers that she's done. And that's a bunch of good information. And I've kind of covered some of these or touched on them, I would say. Also, this is brand new. I just got a newsletter today, and I don't even know if it's on the website, but she's publishing queen breeders that are scoring their hygiene with UBeeO. And I see Mike Palmer is on here. Corey Stevens is on here. And these are in the Northeast, and there's a Midwest, and then it said coming soon, West Coast. So she may be listing, assuming hopefully we have somebody out here. I think Dan Harvey is going to get the kit and be testing with it this year for Olympic wilderness bees. But so this will hopefully will develop and be a resource for us in the Pacific Northwest. So I mentioned Dr. Rinder, and this is his book that he did with Steve Coy. And the reason I like this book is that there's so much of this research. And he was really at ground zero when the mites came in. He was the research leader of the honeybee breeding genetics and physiology lab at Baton Rouge from 77 to 2016. He published like 312 papers. He went to Russia in 1994 and found these Primorsky bees, did 12 years of breeding with them. And so in his book, every chapter has a lot of references. So there's like 113 references that I counted up. So anyone that's interested in what is this hygienic behavior, and kind of getting in on these early studies, this is I find it really fascinating. There's some really cool studies that were done in there. If Van Alphen, this book just came out. I just got it. I'm almost finished with it. And Honeybees, a natural and a less natural history. And he's like I think what you'd call an evolutionary biologist. And he comes at from that angle, and he has some really good information. And he shows the importance of having these feral colonies that are on their own, where they can respond to the diseases as they come, and have like a high availability of these alleles that we lose because of the way the breeding is done these years. If you go to YouTube and search National Honey Show, Van Alphen, he did 2 presentations, and I would say check those out. It's really interesting. And of course, hopefully everybody knows about Dr. Seeley's book, the lives of bees and the, you know, the story of um, the varroa mite and the Arnott forest and, you know, very broad range of information here on how the honeybees live in the wild. So let's to kind of finalize here, get to the end, let's just bringing that page back where we had the characteristics of these high UBeO scoring colonies. Mite levels below economical thresholds, winter survival, better memory of the nurse bees, lower virus loads, mites with lower virus loads, possibly better abundance of the beneficial gut microbes, less nosema, less chalk brood. So what, you know, most of us are, you know, not really satisfied with the treadmill that the industry's been on with these chemicals, and seeing, you know, the mites just develop resistance, and then they bring in a new chemical, and then you know, the chemicals in the honey, and you know, we just want to get away from that. And so, you know, I think what each of us, you know, should do is, you know, what can I do to move the needle a little bit? And I think the either doing your own UBeO scoring, or when you go to buy queens, start asking for daughters from mothers that have been tested. And that, you know, if enough of us were doing that, you know, that's going to move the needle a little bit. And that's it. That's the end. So I have plenty of time for questions if anybody has questions.
Thank you so much, Rex. And I love your graphics. You said
Thank
you
you.
have like training in graphics design too along the way. Picked up some because it's just they're fantastic. These, I guess you did pretty much the graphics in this presentation, and they're
Yeah.
really neat. And people who are going to turn to pumpkins can ask the first question. Okay, it's 8 o'clock, and Rex did a wonderful job of condensing a complex and telling you where to go to get all the extra details that there wasn't time for tonight. So, is anybody going to turn to pumpkin that needs to ask a question? No, I
Are
mean,
they all
because
gone?
everybody has kids, and like I forgot to feed my geese. I had to run out in the middle, so I missed a little bit. I'm sorry. I
Oh,
will watch it
darn.
though
Well,
because it's recorded.
hopefully it's
I
recorded.
probably should stop the recording so it doesn't get too long. So I'll thank you very much, and I will stop the recording now.